All right, everybody. So, good morning or good afternoon, depending on where you are in the United States right now. My name is Jake Massey, and I am a technical sales specialist with Thermo Fisher covering the flow cytometry reagents. Uh, Very happy to welcome everybody to this month's presentation for Let's Talk Flow. Today's topic, we're going to be discussing uh, navigating the optical configurations of your instruments. So we have our presenter today. We have Michael Fuller, who's one of our field application scientists that covers the Southeast region. Um, the the talk today, we're going to walk through just your covering your optical configurations, how light travels inside of a flow cytometer, and why it's important for your experimental design. So thank you all for joining us for our live presentation today. But before we get started, we do have a couple of housekeeping reminders. Your camera, your microphone, and your chat functions have all been disabled. However, if you would like to submit a question for the Q&A, very easy. At the top of your screen, you'll see a Q&A icon. If you click on that, you will see the text box appear where you can submit your questions. And then for any questions that we do not get the chance to answer live, please be assured that somebody from our panel design team will reach out to you with your answer. Also, if you find that you prefer to speak one-on-one with one of our panel design scientists and you have questions and you'd rather have your questions answered this way, we do encourage you to email the team directly with your question at flowpaneldesign@thermofisher.com, and I'll post that link in the discussion field. Um, so we'll just kind of let people come in for another minute or so, and then we will get started. And so in the meantime, we have a poll that should have popped up on the screen. Uh, just simply looking to see how many lasers you have in your flow cytometer. And then we'll hang tight for a minute here, and then we will get started shortly. Alrighty, so we will go ahead and get started. So without further ado, we welcome Mike Fuller to present on this month's Let's Talk Flow. So take it away, Mike.
All right, thanks, Jake. Welcome, everybody. Again, as Jake said, my name is Mike Fuller. I'm a field application scientist for flow cytometry with Thermo Fisher, based out of the US Southeast. And I will be talking to you today about optical configurations of your flow cytometers. So for those of you who were in attendance in our May session, you probably remember talking about how to assign fluorophores and all of the important things that you have to consider when you are choosing fluorophores for your flow panels. Um, those points, if you remember, you need to know how many lasers your flow cytometer has and what the excitation wavelengths of those lasers are. For example, blue lasers are 488 nanometers typically, a green laser could be 532. So it's important to know these wavelengths. It's also important with conventional flow cytometers that you know how many detectors you have available for detecting fluorochromes, as it is a one-to-one relationship with a conventional flow cytometer, and then also what the filters are that are associated with each detector. So ultimately, our goal in picking fluorochromes is to choose fluorochromes that will be as excited as close to their excitation maximum as possible with the given lasers that we have, and where we can capture the fluorescence as close to the emission maximum as possible with the given detectors and filters. And it gives us the strongest possible signal. So today we're going to talk about how we can best achieve that. Remembering back in that previous presentation, we introduced configuration charts for your instruments. These can be very useful. They quickly inform you what lasers you have available on your instrument, how many detectors you have, and what the filters are that determine what wavelength of light can be detected in those detectors. These are really useful if you're picking out fluorochromes. It's important to know which detector they will be recognized in. You can't simply assign them to random detectors. So now let's talk about how exactly we achieve that. So imagine what's happening as a laser hits your cell. All of the fluorochromes that are attached to that cell or that particle that can be excited by that laser will begin to fluoresce. All of that light then is going to come into a detector array. So these can be various shapes and configurations. This is a detector array that we have in our attuned line of flow cytometers. But essentially all of that fluorescent light is going to come into this detector array, and we need to route it to these detectors that are typically around the periphery of your detector array. And in order to do that, we need to be able to distinguish the light coming from the separate fluorochromes. And with that, we use filters and mirrors to split the light up into different wavelengths and send them off to their respective detectors. We identify the shorter wavelengths in the earlier detectors in the array, and then progressively longer and longer wavelengths. So to understand how this light gets split up, it's important to understand how these filters work. So there are four basic types of filters that you will find in most flow cytometers. The first one we're going to talk about is a bandpass filter, frequently abbreviated as BP. These will usually have two numbers following them, first number / second number. What these do is they allow through light within a certain range or band. So in this case, a bandpass 530/30, the first number represents the median wavelength of light that will pass through this filter, and the second number represents the range of that band. So a 530/30 would allow through all light with a wavelength of 530 nanometers plus or minus 15, or half of the total range. All of that light would pass through. Anything outside of that band is going to be deflected off. And so going back to our previous slide, those bandpass filters are the ones that we typically place immediately in front of detectors. So they serve essentially as a gate to only allow through light that would correspond with fluorescence coming from the specific fluorochrome that we want to capture in that detector. We then have long pass filters, or LP for short. These are followed by a single number. The way that these work is they will allow through all light with the indicated wavelength and higher. Anything below that wavelength will be deflected off. Similarly, there are short pass filters, which essentially work the exact opposite of a long pass filter. In this case, a short pass 530 would allow through all light of the indicated wavelength of 530 nanometers and below. Anything above that wavelength would be deflected off. Again, going back to our detector array, these ones can be put in the light path, basically to block out any light noise that we don't want to go any further, or in different types of configurations. They can also be used as collection filters as well. Finally, the last basic type of filter that we use are what are known as dichroic mirrors. These are essentially specialized long pass or short pass filters. So these, much in the same way that in this case, this is showing a dichroic long pass mirror, it will only allow through light of the indicated wavelength and higher. But now any light that is not high enough, or if it were a dichroic short pass, not low enough, will be deflected off at a 90-degree angle. And so we can use these to direct light in a particular direction. So in this case, we can use these directing mirrors to deflect light down through a bandpass filter into the detector. And so using combinations of these filters and mirrors, we can split the light up and send light coming from different fluorochromes to their respective detectors. Now in this slide here, you can see the filter arrays from our Invitrogen Attune NXT flow cytometer that's configured with four lasers, a violet, a red, a yellow, and a blue. And in each of these detector arrays, you can imagine the light path. The light is going to be coming in here from the top, and it's going to pass through here. All of these dichroic mirrors here indicated on a 45-degree angle will deflect light off of different wavelengths, picking up first shorter wavelength fluorescence and then progressively longer and longer ones. And the same thing for all these other ones. And so this allows us to, with a smaller number of lasers, we can still detect fluorescence coming from different fluorochromes. Now, the one detector that you will not see in these configurations typically is the forward scatter detector. We usually use a different type of detector for that, and so it's not typically included in these detector arrays. So with this information, then we can then begin to build our panels and choose fluorochromes. So focusing in here off of this BL3 detector, off of the blue laser, we have this final bandpass filter down here of 695/40. So that will allow through all light of wavelength range between about 675 and 715. And so with that knowledge, we can then find fluorochromes that will work with this. Now, this is really convenient in that we are not necessarily locked into just a single fluorochrome. Frequently, there are multiple fluorochromes that will all work with that same filter. So we have options in case an antibody that you are interested in isn't available in a particular fluorochrome, it might be available in another fluorochrome that would be compatible with that filter. And so that gives you some flexibility in buying your reagents. It's important to know that changing these filters can impact what fluorochromes you need to purchase. So in this case, we've only slightly changed the filter from a 695/40 down to a 670/30. And while technically we might still be able to use these two options that we showed in the previous slide, we now see that by shifting that bandpass a little bit lower, that now the Percy P eFluor 710 fluorochrome we showed before might not be the optimal one. And in fact, it opens up the possibility of trying different fluorochromes. In this case, this NovaFluor Blue 660, now we are capturing its emission maximum much better than we are with the Percy P eFluor 710. So this might be a better option here. So depending on the filter, it can impact what fluorochromes we're choosing. Now, in this case, we've got flexibility. We can choose different fluorochromes if a particular one is not going to give us optimal fluorescence capture. Sometimes, though, we're working with situations where we can't change those. This is frequently the case, for example, if you're working with fluorescent proteins like GFP, YFP, mCherry, etc. There's a lot of those out there. You can't simply change the fluorochrome because maybe the filter set that comes with the flow cytometer isn't going to optimally capture that emission maximum. In those cases, many flow cytometers will allow you to manually change the filters. You can purchase custom-made filters. You can purchase ones that are pre-made. To illustrate this point, I'm going to walk you through using our fluorescence protein filter kit. So we have a supplemental filter kit that you can purchase with our attuned flow cytometers that would allow you to get more optimized detection of both GFP and YFP, as well as mCherry. So with the standard filters, as you can see here, if you had a sample that expressed both GFP and YFP and you needed to distinguish between the two, the standard filter configuration would not be able to do that. As you can see, this bandpass filter here is going to be capturing fluorescence from both GFP and YFP. So you're going to get co-capture of that fluorescence in the same channel. By changing those filters, we can now pick two different filters, one that is optimized for capturing the maximum fluorescence for GFP, and one that's optimized for capturing maximum fluorescence of YFP. And that now allows us to look at expression of both of those in the same sample. Similarly, for mCherry, the standard filter, it will capture mCherry fluorescence, but it's not quite capturing that emission maximum, which you might need if whatever cell this is expressed in, it's not expressed at high levels. So by changing that filter, we can now capture that emission maximum, and that will give us better detection capabilities, particularly if you're looking for rare events or for markers that are not very highly expressed on any given cell. Coming back to our instrument configuration chart, again, this is a very useful tool for helping you determine which fluorochromes you can potentially use in any of your given detectors. However, this is not an exhaustive list. We're not printing these every other day to include all of the newest fluorochromes that are being created. So while this can be a useful tool, we also highly recommend that you use spectra viewers, which can be found online. Thermo Fisher has a spectral viewer. You can do a search for that. That allows you to put in your specific instrument configuration, what lasers you have, what bandpass filters you have, and then you can test out different fluorochromes and find which detection channel would be optimum for detecting that particular fluorochrome. Now, up until now, we've been talking about conventional flow cytometers. And what I mean by that is flow cytometers where each detector is tied to detecting fluorescence from one fluorochrome. So it's a one-to-one relationship between fluorochromes and detectors. So if you have a flow cytometer that has 10 detectors, you can build up to a 10 color panel. There's also out there spectral flow cytometers. Maybe you're using one or you've heard of other people talking about these. The way that these differentiate themselves from conventional flow cytometers is that rather than looking at fluorescence for a fluorochrome in just a single detector, we now detect the total fluorescence over the entire spectrum. So every fluorochrome will be detected in all channels. What this does is it gives you the opportunity of looking at more fluorochromes with fewer lasers. So while it does expand your capacity for building panels with more and more colors, there are some important things to realize with these. One of them is that you still need to be mindful of what your optical configuration is. So this is showing the detector array from our Bigfoot spectral cell sorter for one of the lasers, the 405 nanometer laser. And if we look in close here, we can still see that we're using the same types of filters and mirrors in here. There's bandpass filters, there's long pass filters, there's dichroic short and long pass filters. So we're still directing light to the different detectors in the same way that we are with other ones, except in this case, we're detecting total fluorescence over the whole spectrum for each fluorochrome. Now, while that does open up possibilities, this configuration can still greatly influence the ability to distinguish one fluorochrome from another. To illustrate that point, we're going to focus in here on the detector array for the 349 nanometer laser and focusing in on this particular detector here. So with that detector, we're going to capture the emission maximum from both brilliant ultraviolet 805 and 737, as you can see here. Now, given the number of detectors and filters that we have here and how close these are together, we might not be able to well resolve these two fluorochromes from one another. However, if you had a different cytometer with a different number of detectors, maybe there were more detectors in here and more filters, you might be able to better resolve that. So while these spectral instruments will give you more possibilities for fluorochrome combinations, it is still important to know what your filter configuration is, know how many detectors you have, and that will highly inform which fluorochromes you can and can't use in a given panel. So just to summarize, it's really important when we are selecting fluorophores for our panels to know your instrument's optical configuration. This includes knowing how many lasers and what kinds, how many detectors you have available, and what the filters are that determine the bandwidth of light that can be detected in those through those filters. Depending on the instrument, if you need to stick with certain fluorochromes and you cannot alter that, they can be flexible by being able to change out filters to better capture the emission maximum. Um, While instrument configuration charts can be very useful in selecting fluorochromes, do not dismiss using spectra viewers. They will give you much better information on both how optimally a fluorochrome will be excited and what detector will best capture those emissions. And finally, even though we have more freedom with spectral flow cytometers, you still need to be mindful of those same rules. You still need to know how many detectors you have and what your filter configuration is. Thank you very much for attention, and I'll turn the time back over to Jake.
All right. Thank you, Michael. Appreciate it. So while we have everybody's attention, we also want to mention our panel design service as well. So this is a completely free service that Thermo Fisher offers to work with designing your panel from any stage of the panel design process with an expert. It is a totally personalized design with a one-on-one customer assistance program that we offer.